Let's Talk About Pain, a podcast from the Quebec Pain Research Network. In each episode, we discuss the latest advances in pain research with guests from across Quebec in the health and research communities. Enjoy your listening.
Hello and welcome to another episode of Let's Talk Pain, created in collaboration with the Quebec Pain Research Network. My name is Hannah Derue, and I'm a Ph.D. student at McGill University under the supervision of Dr. Alfredo Ribeiro-da-Silva and Dr. Jeffrey Mogil. Today, I'm going to introduce you to Dr. Jeffrey Mogil, James McGill Distinguished Professor in the Department of Psychology at McGill University, Canada Research Chair in Genetics of Pain, and E.P. Taylor Chair in Pain Studies. We'll talk about his background, his expertise in research, as well as his perspective on chronic pain management and how we investigate it. Hello, Jeff. Could you please introduce yourself and the area of your work?
Hi there, Hannah. My name is Jeff Mogil. As you mentioned, I'm a pain scientist here at McGill. And our lab studies pain from a wide variety of perspectives, mostly skewing to the basic science end of things.
So where does your interest in pain and pain research come from?
Well, as these things usually go, it happened entirely by accident. When I first got interested in science, I was doing sort of reward and reinforcement work, things related to drug abuse. And, one thing led to another, and suddenly I was doing pain. I rationalized it at the time. I said, pain, pleasure, two sides of the same coin. Interestingly, I haven't really brought those two things together in my lab over a 30-year career until you and the research that you've been doing in the lab for the past year.
Well, great, and I think we're finding some interesting stuff, but maybe we'll talk about that another time.
I think so too.
So I wanted to ask, we'll talk obviously about what you're doing now, but I wanted to know if you have a past researcher or someone that you've looked to for inspiration.
Well, that's a hard one, mostly because I think there's way too much hagiography in science already. Science isn't really about people, it's about data and findings and systems. Now, of course, yes, people do science, and one can be very impressed with scientists if you want, although in my mind, people are way too impressed with individual scientists, and they forget, first of all, that science is a team sport and that lots of people at lots of different levels with lots of different functions are involved, and it's hard to say that any one of those is more important than any other, even though it's people like me that end up getting the credit. And I also strongly believe that science involves a lot more luck than anyone is willing to admit or wants to admit, which means that people that are successful, maybe they're very smart and maybe they're geniuses and maybe they just got lucky, and it's really hard to tell the difference. And I don't think it's worth spending too much time worrying about that sort of thing. Obviously, I have scientists who I look up to who have been very important as, role models for me earlier in my career. People like Alan Basbaum and Frank Parecka come to mind. But again, I just don't think thinking of science in that way is particularly accurate or productive.
So I'll follow up on that question, if you'll allow me. If it's not people that we look towards or other scientists, where do you look when you're looking for inspiration for new research questions?
Well, that's a really good question, and that's also a hard one. Sometimes you go to a meeting and someone says something and that gives you an idea, right? A lot of science is just analogy, right? They did this for that, so I can do this for what I do. If it worked for that thing, it might work for my thing. And a lot of science just comes down to that. Mostly, scientific ideas are born from the data you've just collected, right? So you collect data for one experiment and things may not go exactly like you predicted. if they did go exactly like you predicted, like that's great, you're going to publish a paper, but it hardly gives you a new idea. But when things don't go exactly as predicted, that's when the new ideas come, and the best ideas. And so, when I look back on my career and I see the papers that have made the biggest impact, all of those were sort of accidents that we were led to simply by taking a long, hard look at the data we were collecting and noticing subtle things that, led in new directions.
So for those listening, the key to being a good scientist is making many, many mistakes.
Many, many mistakes and many, many graphs. You need to slice and dice your data in every single way. I'm a big believer and a big fan of looking really hard at data in a number of different ways, and that's when new ideas gel in your mind.
So there's a huge gap between people who do what we do, preclinical research, and clinical researchers or the general public. How do we find a way to have our information that we gather in all these experiments reach the real world?
Well, that's an interesting question. I actually believe that we worry about that too much. The idea of knowledge translation for basic science, sometimes our findings deserve to get to the public. And, a lot of the findings that I've made over the years have gotten to the public because they're interesting or funny. In some way. So, when you publish a paper saying that mice are afraid of men, that's the kind of thing that the public will end up hearing about, because when people hear that statement, they chuckle. But a lot of times we publish findings. For example, in the early part of my career, I provided evidence that a lot of genes were involved in pain that no one knew about before. And frankly, between you and me, a lot of those papers were of interest only to 20 other people in the world, right? It was extremely important, however, that all 20 of those other people found out about it. And of course, they will find out about it because papers are published and I go to meetings and talk about it. I mean, there are systems in place. Now, does the public really care whether it's gene X or gene Y that's involved in pain? No, they don't, and they shouldn't. They really, really shouldn't. The things that the public needs to know are when scientists are very, very sure that something is true. And only then, when there are a whole bunch of clinical studies and meta-analyses of those studies, and even better than that, meta-analyses of the meta-analyses, that's when the public should know things. Before that, it leads to hype, which then leads to a backlash. And sometimes I think we go too far in trying to make the public aware of everything we're finding. That being said, the public does need to know something, lest they think that basic science isn't needed. The public in some way needs to be reminded that all of the clinical research that they care deeply about because it's very close to them, or at least closer to being useful than, what we do with mice. The public needs to remember that that clinical research wouldn't have existed without the basic research that came before it. And so clinical research that happens today is only occurring because 20 years ago someone did basic research. And if anyone wants there to be clinical research in 20 years from now, then there's going to have to be basic science today. And so I think we all have to be grown up about this and remember that in addition to having a responsibility to people in pain today, we also have a responsibility to people in pain 20 years from now, and 50 years from now, and 100 years from now, and 1,000 years from now. And the only way those people are going to be served is by basic science today.
Okay, I think this actually goes really well into my next point. Research is really, really valuable. I think we both agree as people who do it that it's a good thing that we do. But it's incredibly resource intensive. It takes a lot of time, a lot of money, and a lot of labor. So one of the things that I'd want to ask you is if you had the chance to sit down with a political figure or someone who has an ability to convince the public that this is really useful and that we should invest more in it, what is your argument that you would give them?
Yeah, it's a big challenge because there's a fundamental disconnect with the priorities of politicians and what I believe is true based on what I just said, right? So politicians are elected on 3 or 4 or 5 year mandates. That's their time horizon. And what they're very interested in is what can be accomplished before they get out of office. So hopefully they can get reelected for another term. But what I'm talking about is the long game, right? The reason we're doing basic research isn't so anything gets to patients in pain 3 years from now or 5 years from now. That's not realistic, right? The timelines involved are much, much longer than that. And so what I wish that people understood and what I wish that people could get their head around is that they need to have patience, because we are indeed playing a very, very long game. And we've been playing this long game for a long time, and it does pay off. The reason why people are living longer than they ever had, and the reason why there are treatments for things and cures for things today, is because people started doing research decades ago, and they are the beneficiaries of what's happened in the past. If people in the past did that for us, we need to be ready and willing to do that for people of the future. Abdicating that responsibility would be just disastrous in my mind, and just utterly unethical. You mentioned that science is resource intensive, it's expensive, it takes a long time, it takes many people to do it. Another thing I'd like to remind everyone of is that it's also incredibly inefficient. In fact, it's the most inefficient thing people do, right? If we knew what we were doing, it wouldn't be science, it'd be engineering. The very fact that it's science means we don't know what the answer is, and we're going to look down a lot of blind alleys, and they're mostly going to be blind alleys. That is the nature of the beast, right? There's 22,000 genes, there's maybe 40 or 50,000 proteins. Only a few of those are critical for pain. And the only way we're going to find which ones those are is by going through and testing them one by one, laboriously. Most of the time, those proteins actually aren't going to be very important for pain, and the whole thing is going to lead to nothing. But somewhere out there, and I believe this very strongly, there are proteins that will lead to drugs or other sorts of interventions that are hugely important for pain, and one day some lucky or clever soul, but probably lucky, is going to run into the right one, and that is going to result in some breakthrough treatment for pain. We just have to be patient enough to wait until that happens. And of course, we have to keep going, because if we don't keep going, no one's ever going to find it.
I think I'd like to ask, looking towards the future of both chronic pain research and clinical practice, but specifically research, just going off of the point that you made, how do we make research more efficient in 100 years from now?
I mean, part of the answer, of course, are robots and AI. I think people have overstated the utility there. I recently saw Sam Altman, the CEO of OpenAI, give a talk, and he was sort of claiming very strongly and very confidently that AI was going to solve cancer within a couple years. And of course, if AI can solve cancer, it could also solve pain. And I think either Sam Altman knows but decided not to say, or maybe actually doesn't know that that's not the way cancer research, or pain research for that matter, works, right? It's not a logic problem that we haven't solved yet. It is exactly what I suggested before, a rigorous and methodical search one by one through all the possibilities. And of course, as you know, searching through all those possibilities requires that you make a particular mouse, and you have to test that mouse, and you have to breed that mouse, and mice only breed as fast as they breed, and AI isn't going to solve that problem for us. Now, AI is definitely helping. It's letting us do certain things faster than we did before. Maybe AI is going to eventually have better hunches than I do or you do about which molecules or brain areas are the right ones versus the blind alleys. I'm not claiming that it's not going to help, but I have a feeling that what we do is fundamentally a slow manual process that is going to continue to take a lot of time and a lot of resources.
Actually, if I can follow up on that, much of AI is based off of developing programs based on what you put into it. As programmers say, garbage in, garbage out. With the reproducibility crisis, does that also add a layer of complexity to using AI for research?
Oh, yeah, but this is a question that's big and above my pay grade, right? I mean, there are many people that are starting to believe that AI is starting to eat itself because they're running out of training data. And so to give AI more training data, they're using AI to produce the training data, and the GIGO problem just gets bigger and bigger. But again, that's a software engineering question. I only know what I read.
In the end, I think you're right about saying that AI is as limited as the research that we're able to give it. If we make good research, the AI can help us, but only if we produce it.
Right.
One last thing. With these advances and moving forward in the future, do you think that we'll have a new revolution in clinical care for patients? Or do you feel that we just have to continue slogging, as you mentioned, and continue trying our best? What do you think is the next big advance in 100 years from now for clinical care?
I mean, I think it's both. I think we're going to continue slogging, and one day, out of the blue, someone is going to find something that is broadly effective. People talk about magic bullets, and magic bullets have a bad rap. But you know what? Magic bullets exist, right? Antibiotics are a magic bullet. Knee replacements are a magic bullet. There are magic bullets in medicine. There aren't many of them, granted, but they definitely exist. And I see no reason, theoretically, why there isn't a molecule that can either be blocked or enhanced somewhere in the brain or the immune system that would be broadly efficacious against pain. Now, we haven't found that molecule yet, but that doesn't mean it doesn't exist. We've only barely begun to search.
Well, it's really interesting that you bring up antibiotics, because if anybody was lucky in finding something, it was definitely Pasteur. Yeah. So maybe we have another Pasteur in the wings, and right. Someday we'll find the magic bullet for pain.
Someone's going to do it one of these years/decades. I'm convinced of it. It's a biological system after all. And the fact that it's subjective, pain is complicated, as you know. It's not like cancer. Cancer's child play compared to pain for a number of reasons that we could talk about that you're probably well aware of. But at the end of the day, it's a biological system like any other. And biological systems can be unlocked because some of them have. And I expect in time, all of them will.
Well, I think this is the end of the episode. Do you have anything else to add before we wrap up?
No, except that we went in some interesting directions here. I enjoyed this conversation very much.
I did too. Thank you.
Thank you for listening to Let's Talk About Pain. Don't hesitate to subscribe so you don't miss an episode, and to share the podcast with your friends. For more information, visit our website at QPRN.ca or our YouTube channel by searching Quebec Pain Research Network. See you next time.